A study was conducted to study the efficacy of different concentration of different garbage enzyme towards growth performance of spinach, which is Amaranthus species. Amaranthus is a cosmopolitan genus of annual or short-lived perennial plants, collectively known as amaranths. In this study, garbage enzyme was prepared from various media and applied to Amaranthus species as a supplement. Amaranth was grown for 8 weeks and the garbage enzyme was applied every 3 days for 2 weeks. Plants were grown for 8 weeks before being harvested. Fruit peels, vegetables waste, fruit and vegetables are the types of garbage enzymes that apply at different concentration, which is 0, 10, 20, 30, and 50%. To determine the growth performance of Amaranthus species in the parameter of plant height and stem diameter. In short, garbage enzymes are expected to improve the growth of Amaranthus species, but the exact types and concentration need to be determined by carrying out the following studies. Throughout the mini project, the software used for data analysis is SPSS and Microsoft Excel. The tool we will be using to observe aggregate variability found inside sets of data is analysis of variance, ANOVA. In this study, two-way ANOVA is used to determine whether there are any statistically significant differences between the means of 3 or more independent groups of data. Post hoc analysis also required in order to distinguish which group is actually different from each other when they are significantly different exists in between the groups, but ANOVA does not tell us about it.
First, the data is organized using Microsoft Excel according to types of garbage enzymes, concentration of garbage enzyme, plant height, and stem diameter. The information is then compiled and simplified into a graph that compares the means of dependent variable among the independent variable. After that, the graphs are examined. These Excel-based data are now prepared for export to SPSS for additional comparison and analysis. Next, we are comparing plant height and stem diameter in response to the concentrations and type of garbage enzyme, and the first method that we use is normality test, which determine if the data were normally distributed or not. For this test, the null hypothesis is the mean of plant height and stem diameter is normally distributed, and the alternative hypothesis is the mean for plant height and stem diameter is not normally distributed. If the p-value is less than 0.05, null hypothesis is rejected and data transformation is required because there are less than 30 means. The assumption of normality is accepted if p-value is greater than 0.05 and the next step can be performed. Since the total n in this mini project is 75, which is greater than 30, we can continue the parametric test with bootstrapping. Then, Levin's test for homogeneity of variance will be conducted. The null hypothesis is the variance of plant height and stem diameter was homogenized, and the alternative hypothesis will be the variance of plant height and stem diameter was not homogenized. The outcome of the Levin's test has a significant impact on how the data will be analyzed subsequently. Next, we run a statistical test called Analysis of Variance, or ANOVA, which examines how the various groups vary from one another and the p-value will be analyzed. Lastly, it is necessary to conduct a post hoc test to determine which means significantly differ from one another if null hypothesis of the ANOVA test is rejected. A Tukey test can be used as a post hoc test if null hypothesis of Levin's test of variance homogeneity is not rejected.
Before we continue with parametric test, we shall test normality of data. So as in 3.1, so both plant height and stem diameter are having p-value that less than 0.05 of our alpha value. So null hypothesis rejected. So both plant height and stem diameter, they are not normally distributed. However, since we are having sample number of more than 30, which is 75, so we can perform bootstrapping of data as in 3.2. Before we proceed with ANOVA, so we shall test the homogeneous variant to fulfill the criteria for ANOVA. So as in 3.3, both plant height and stem diameter are having p-value that is larger than our alpha value 0.05. So null hypothesis failed to be rejected. So both plant height and stem diameter are having homogeneous variance. So as in 3.4, the ANOVA of type of garbage enzyme and concentration of garbage enzyme effect on the plant height and stem diameter. So both are having the p-value of smaller than alpha value 0.05, so the null hypothesis is rejected. So there's at least one group of mean is different from the overall mean. Since there is significant difference in the ANOVA test, so we'll continue with Turkey post-hoc test on type of garbage enzyme and its homogeneous subset. So all garbage enzyme of plant height and stem diameter are having p-value that is smaller than the alpha value 0.05. So null hypothesis rejected, and thus the effect of each garbage enzyme is different from each other. Then 3.6 will be the Turkey post-hoc test on the effect of concentration of garbage enzyme on the plant height and stem diameter. So they are all groups were significantly different from each other except the pair of 30-50 in plant height, whereas the other 10-30, 10-50, and 30-50 in stem diameter, they are not significantly different. So there are four homogeneous subsets are obtained from the post-hoc study of the concentration of garbage enzyme. Table below shows the homogeneous subsets of plant height and stem diameter of the effect of concentration of garbage enzyme. Since type of garbage enzyme and concentration of garbage enzyme does affect the plant height and stem diameter, so we conducted the factorial factor to study the relationship of the garbage enzyme and the concentration. So we have done the ANOVA of factorial factor on both plant height and stem diameter. So ANOVA table of plant height and stem diameter are having a p-value of smaller than 0.05, so the H now is rejected, and thus there is a significant difference in the mean of plant height and stem diameter of the factorial factor. Based on the Turkey post-hoc test of factorial factor on plant height, at homogeneous subset can be obtained with the highest of mean is done by 20% of food and vegetable waste. Based on Turkey post-hoc test of factorial factor on stem diameter, so they are at homogeneous subset obtained as well, and the one with the highest mean is from on the 20% of food and vegetable waste. So that's all from the results part.
The graph was plotted from the data collected. We can see that the graph of plant height and stem diameter against garbage enzyme concentration, there is a general increasing trend from 0% to 20% of garbage enzyme concentration, whereas the concentration after 20% there is a decrement of plant height and stem diameter. As for the graph analysis of plant height and stem diameter against type of the garbage enzymes, we can see fruit peel and vegetable waste garbage enzyme had the similar effect in affecting the plant height, whereas mixture of fruit and vegetable waste enzyme shown a higher effect on increasing the plant height. Moreover, from graph 2, all three types of garbage enzyme are having similar effects on the growth of stem diameter, as we can see from the graph that there is almost same level of mean stem diameter for all three types of enzymes. As for plant height, we can see the lowest effect on plant height are 0% fruit peel, 0% vegetable waste, 0% fruit and vegetable waste, 50% fruit peel, 30% vegetable waste. As for the factor that has higher effect on increasing the plant height is 30% of fruit peels. As for the stem diameter, we can see the lowest effect on plant height are 30% fruit and vegetable waste and 50% fruit and vegetable waste. As for the factor that has higher effect on increasing the plant height is 30% of fruit peel. As a result, the plant receives an optimum enzyme concentration for its growth, which is 30%. This optimum enzyme concentration mitigate the plant hormone that increase in cell division of the plant. Too low and too high concentration of enzyme will inhibit the growth of the plant. As the data was not normal and having a higher density, thus we perform ANOVA with bootstrapping. Then the null hypothesis is rejected, so there is a significant difference of mean between the plant height and stem diameter. Since ANOVA showed there is a significant difference of the data, thus post-hoc test using Turkey HSD has been done. Based on Tukey multiple comparison, all three enzymes are significantly different from each other. As for the ranking of plant height, the lowest is fruit and vegetable waste, and lastly fruit peels. As for ranking of stem diameter, the lowest is fruit peels, followed by vegetable waste, and lastly the highest data fruit and vegetable waste. As for Tukey post hoc test of plant height, we can see that the 0% garbage enzyme concentration is in a homogeneous subset, and 50% and 30% garbage enzyme are on the same subsets, and 30% of garbage enzyme is on a different homogeneous subset. As for Tukey post hoc, the test of stem diameter, we can see that 0% garbage enzyme concentration is in homogeneous subsets, and 10% of garbage enzyme are on a same subset. 10% and 30% of garbage enzyme is on a same homogeneous subset, whereas 30% of garbage enzyme is on another different homogeneous subset. So we are having two factors to determine the effectiveness of both type of garbage enzyme and concentration of garbage enzyme on plant height and plant diameter. Stem diameter. A multiplication of two factors is done to conduct a two factorial of ANOVA based on the ANOVA result. It shows that there is significant difference between plant height and stem diameter when factorial ANOVA is done. Since there is significant difference between factorial independent variable on plant height and stem diameter, the Tukey post hoc test is done to study the relationship between two independent variables. That's all from